Thanks very much. It's a pleasure for me to be here. And what I want to talk about specifically is an approach that we've been developing over the last, if I stand here, you can hear me okay? I can be a lot louder, too, if somebody starts to doze off in the back. That we've been developing for treatment of tumors in various places in the body, but I want to talk particularly about treating intracranial tumors today. And I want to present this sort of historically as a story that started at the time that I was a faculty member at Johns Hopkins. For full disclosure, I'm a scientific co-founder and a consultant for a small biotech company here in town that specializes in nanoparticles for delivery of siRNA. But they've not supported any of the work I'll be talking about today. So the general theme is using materials, in particular polymers, as tools for the delivery of medicines, in particular chemotherapy agents here. And so you all probably know what polymers are. They're long chain molecules. This is a linear polymer chain formed by covalently coupling monomeric units together to form stable, long molecules. And because of their properties, in particular the large size of the molecules, they can form materials that are quite stable. And because they can be produced relatively inexpensively, they're widely used in all sorts of aspects of our lives. We've come to appreciate and know plastics or polymers as materials. They entered the cultural aspects of our thinking many years ago. And one thing that you can do with materials like this, with certain kinds of polymer materials, is fashion them into devices that can be used in the body. So this is an example of a material that's both biocompatible. It's a polymer that if you implant it in tissues, it provokes very little response from the tissue around the implant site. And it's biodegradable, and that it's been designed of polymers that have structures that look like this, but that the linkages between the molecules are unstable in water or unstable in the body. And so the molecular weight of the individual chains, after it's implanted, slowly decreases, and eventually the device itself just dissolves. In addition to having degradable linkages, the polymers I'll talk about are made of monomers that are non-toxic as well. In fact, I'll principally be talking about polymers that are made of lactic acid and glycolic acid, which are natural metabolites. So what you end up with after some months after implantation is nothing, hopefully. This polymer would ordinarily be clear. You could see through it. It's opaque in this case because it's been loaded with a chemotherapy drug. In this case, it's the nitrosourea, BCNU, and loaded at about 4% by weight of the polymer is the drug. So there's so much drug that you can't see through the material anymore. And if you took this kind of material and you dropped it into a test tube full of water, then one of the properties we've been able to engineer into materials like this is the ability to endow them with a slow release of the drug. So this just shows that simple experiment where you take this material and you drop it into water, and then you measure in the water how much of the drug has come out of the device as a function of time. And this shows a typical example where one can get many weeks of release of the drug at a fairly reliable and continuous rate just by applying a single wafer. So the first application that we worked on here, and again, this was work that I started when I was an assistant professor at Johns Hopkins, was to explore whether one could use these materials for treatment of brain tumors. And I don't have to tell you that brain tumors are very difficult to treat with chemotherapy because you deliver systemic drugs. Very few of those molecules get into the brain, usually cause toxicity systemically before they cause any effect. And so the idea, and it's also known that from long experience with surgery of brain tumors, that if you surgically resect a tumor, they'll often recur. And many times they reoccur at the same site where the tumor was removed. So in many cases, it's a local disease. So it could be treated perhaps by pharmacological interventions locally as well. So the idea was to develop these materials as a tool that neurosurgeons could use. At the time of surgery, when the resection was complete, they would then line the resection cavity with these wafers. And The patient would then leave the operating room with these devices in their brain. Over the next few weeks after the operation, the drugs would be slowly released. They would be available to treat disease locally at the resection site. And then after the drug was released, the polymer wafers themselves would dissolve. So this approach, which I worked on with a team of people, including Henry Brem, who's chief of neurosurgery now at Johns Hopkins, and Bob Langer, who was my mentor at MIT, was approved by the FDA in 1996. The clinical product is called Gliadel, and it's used around the world as an adjunct to surgical treatment of tumors. And this is just an example of a tumor that's been removed, and the dark spots here on MRI are wafers that are inside the brain of a patient who's receiving this therapy. So since that time, we've thought about whether one could sort of do better, whether you could make these things even more effective at treating tumors, and develop approaches that would maybe be useful even in the absence of surgery, that could be delivered without a major surgical intervention like this one requires. And so the first step of that was to make these materials smaller, and this shows about 1990, the early 90s. We were making microparticles about a micron or so in size, so about the size of a bacterium, loaded with drugs. This is made of that lactide glycolide copolymer I mentioned before, a material that's well known to be safe in clinical practice. And these are loaded with drugs, and you can achieve some of the same kinds of things I described with those wafers. If you put these into water, the drug molecules are released over a period of time that can extend for many weeks or months. And we learned how to engineer these materials so that one could control the rate of drug release, so you could make tiny particles that released at whatever drug at whatever rate you wanted. It took us about 6 or 7 years after that to decrease the size by about a factor of 10. These are particles that were made by Amaryllis Sanchez at Cornell. She was a graduate student who worked with me there. These are particles that were unique at the time for a couple of different properties. One is that they're quite small. These are 160 nanometers in diameter, so roughly the size of a virus now. And they're fairly uniform. They're small and discrete particles that can be easily resuspended in solution. And what was most impressive about these, because there are lots of ways to make tiny particles, but there's very few ways to make tiny particles that are loaded with large quantities of drugs. And these contained up to 20% by weight camptothecin in this case here. So one can make particles that are this small, and one can load them at high levels with drugs. And if you incubate them in water again, then you see the drug molecules come out of the device remarkably slowly, considering how small the particles are. One interesting feature of being this size is that these particles, being less than 200 nanometers in diameter, are much smaller than tumor cells. So these are breast cancer cells in culture. And if these particles also have the property that they're fluorescent, because the camptothecin that's within them is fluorescent. So if you incubate these particles with cancer cells, what you'll find for many different kinds of cancer cells is that the particles relatively rapidly enter the cells, and they fill up the cytoplasm of cells like shown here. You can see that the cytoplasm in these cells is fluorescent because they've taken up large quantities of these particles when they were just added to the culture medium. So nothing special done there. If you do this at a low temperature, then very little particle uptake happens, so it appears to be an endocytic mechanism. But the particles are relatively rapidly escaped from endosomes, and you can find them dispersed throughout the cytoplasm. So that might be a useful feature in that you can then take these particles, which are loaded with drugs, and put them inside cancer cells, so they'd be releasing drug from the inside near the target. In this case, camptothecin, the target is topoisomerase. The particles themselves aren't toxic. You can add fairly large quantities of them to culture, and you see no change in viability over time. If, on the other hand, you add particles that are loaded with a drug like camptothecin, and these were experiments that Amaryllis did with Sarah Rockwell, you find these are cells that are sensitive to camptothecin, so you add camptothecin in culture. The green line here just shows the dose-response curve for the free drug. When we added the drug encapsulated in nanoparticles, you see a shift to the to the left of the dose response curve, so they're active at a lower concentration. And this was initially surprising to us because remember that these are particles that have a lot of camptothecin in it, but it's released relatively slowly over a period of a week or two. And this was an experiment that only allowed a 24-hour incubation with the particles. And so most of the drug is presumably not free and available to be active. It's still in the particles. And yet, even though that's true, the particles are more effective on a per-dose basis than the drug itself, we think, because it's releasing on the inside, and so drug's able to accumulate intracellularly to higher levels. To test whether this in vitro property would translate into an animal model, Amaryllis did these experiments again with Sarah, where she inoculated tumor cells in the flank of mice, waited until it was at a fairly aggressive stage of growth, and then tried to treat the tumors in a variety of ways, including conventional treatments with camptothecin and treatments with nanoparticles, and then just followed tumor size over time. This shows a couple of different experiments. In most cases, no matter how we delivered the camptothecin or the particles, you would find that you could slow down tumor growth in the short term. So all the treatments were done at day 10 here, but that eventually tumor would start to regrow again. In sharp contrast, in one particular set of animals, the animals where we directly injected the nanoparticles into the tumor, we saw that no further growth occurred, even after only injecting a single dose of the camptothecin-loaded particles at day 10. So how do you apply this in the central nervous system? So the challenge there is getting the particles where you want at a tumor site. And so in concert with this, we've developed approaches for doing convection-enhanced delivery. That is by putting microprobes or catheters into the brain, infusing under relatively high pressures so that you create an interstitial fluid flow that will carry drugs away from the probe tip and into the brain. And this just shows some early experiments. This is with a microfluidic device that you can place in the brain. You can actually go up to fairly high pressures, and because the device was so small, you get very little backflow of fluid up the channel. All the particles are deposited, or the drug is deposited right around the tip of the injection site. And you can control how far the particles go by adjusting the pressure and flow characteristics through the microprobe. So the idea was that people had used this approach before for infusing drugs, and you can do that. You could infuse just drugs into the brain here, but most drugs have a relatively short half-life, so you do an infusion, and then in a relatively short period of time, let's say an hour or several hours, most of the drugs has disappeared by that point. So your option is either to do continuous infusion or re-infusion, which obviously can be difficult. Instead, what we proposed was that you could infuse these nanoparticles locally, that they'd be small enough to penetrate away from the tip of the microprobe, and that they would hold on to drugs and release them slowly. So if one went back even days or weeks after a single infusion, you should still find a drug right around the site where it was infused. And in fact, in early experiments that were done by Andy Sawyer, a Yale PhD student, and Keith Neves, Cornell PhD student, they showed that you could deposit nanoparticles, and we had some control over where these particles were deposited within the brain. If you load those particles with camptothecin, inject them, again, a single injection, again at day 10 after implanting a tumor. This time it was an intracranial tumor. And then just look for animal survival as a function of time. Animals don't survive for very long with these intracranial tumors. If you treat them just with camptothecin, a few animals, or actually just one animal in this cohort, would survive for a long time, but not much difference in mean survival. But if you load the nanoparticles and infuse, then you see an increase in mean survival and a significantly greater number of animals that are surviving. Now, the problem with this technique, we were pleased that it worked, but the problem that we knew was that even though we were infusing these directly into the brain, these particles were about 120 nanometers in size. The interstitial spaces in the brain are significantly smaller than 100 nanometers, and so it was wishful thinking on our part to think that particles were going to diffuse far enough into the tissue to be able to treat all the cancer cells. Still, it works. We think it works because there is some penetration of some particles, and they carry enough dose with them over time that you do get an effect. But in these early experiments, we were seeing volumes of distribution in the brain that were only about 4 cubic millimeters. That's shown here. This is in a coronal slice of the rat brain. Here's the midline. And so you can see the corpus callosum here, so you get an idea, if you're familiar with rat brains, about how small this volume of distribution is. Zhangbing Zhao, whose wife had a baby yesterday, and Toral Patel, who's a neurosurgical resident, have worked on a couple of different strategies for making this better. One is by making the particles smaller, and now these particles are on the order of 50 to 60 nanometers, and so quite small. And improving some of the details of infusion techniques, you can markedly increase the area of coverage of the volume of distribution of these particles. And so we're encouraged that through further engineering of particles like this, we'll be able to treat fairly large-sized intracranial tumors with this method. These particles I've described so far are what we would call naked. They have drugs loaded on the inside. The orange is this polymer that holds everything together. The green is supposed to indicate drug molecules, but there's nothing on the surface, just polymer. And even these fairly simple systems, because of their small size and because of the work we've done to learn how to put active agents inside them at high doses, can be used in a variety of different settings. And so I just wanted to show you some of the things that we've been working on over the past few years. You can load these with plasmid DNA, and they will be then gene delivery vectors. And we're using these in experiments with Dan DeMeo and Alessandra Santon now to study whether this can be a useful approach for treating cervical cancer and ovarian cancer. You can load these particles with siRNA, and they're remarkably effective at delivering siRNA both in cultured cells and in animals. You can, in recent experiments with Peter Glaser, we've loaded these with peptide nucleic acids that form triple helixes and donor DNAs, and they can lead to long-lasting genomic recombination in cells. And we've developed some novel polymers that have never been synthesized and tested in animals before. And some of these particles are highly hydrophobic, and they appear to accumulate more in tumors after systemic injection than the normal polymers do. And so we're exploring that as a way to sort of use this for systemic injection as well, particles that would accumulate within tumors. But I think one of the most promising aspects of this is that we could not only load particles highly with drugs or combinations of drugs, but that you could also decorate the surface. And we had a breakthrough in this area in about 8 years ago. Tariq Fahmy, who's now an associate professor in biomedical engineering, was a postdoc who worked in my lab, and he developed this approach for decorating the surface of these particles with avidin. So you would palmitylate avidin, make it amphiphilic, and so it assembles on the surface of the particles. And the advantage of doing that with avidin is that avidin has 4 binding sites for biotin, and so then you can add any biotinylated substance you want in solution, and it will assemble on the surface of these particles. And so you could add antibodies, you could add molecules that provide sort of stealth properties that make it resist absorption of proteins and disguise it. You could add peptides that enhance uptake. And even adding all these functions, these are still quite small particles. I show fully functionalized particles here. They're still about 120 nanometers in diameter. Just one example of this is work we did with Rick Matthews when he was here in neurobiology. These are particles that have CD44 antibodies that bind to CD44 on the surface. You can see if you put the antibodies on the surface of the particles, it makes them bind avidly to CNS1 cells, whereas the naked particles basically ignore the cells, so antibodies could be added for targeting. You can add cell-penetrating peptides. This is a peptide derived from Antennapedia, known as penetrin, and if you add that to the surface of particles, you can make particles enter cells that ordinarily they don't enter very readily. So these are in endothelial cells that will take up particles very slowly on their own if they're naked particles. But Kim Woodrow showed that if you add enough penetrin to the surface, that you can dramatically enhance the rate and number of particles that are taken up by endothelial cells. And Chris Cheng, who's a PhD student now in MBMB, has developed different approaches. You might be asking if there's a problem with having particles that have high amounts of avidin, of foreign protein, on the surface. And avidin is used in pharmaceutical preparations. We don't think that that's particularly a problem, but you can avoid the avidin altogether if you want by just taking the molecule of interest, and here is the example showing a peptide and coupling it through a fairly common conjugation chemistry to a phospholipid, DSPE. Now this phospholipid peptide conjugate has that same property of being amphiphilic, and if applied in the right way during fabrication will assemble on the surface. So you've got the particle that's decorated with fatty acids that are conjugated to whatever peptide you want, and the particles work the same way after you've added this coating. Just to show how they work, these are prostate cancer cells, KB cells, that overexpress folate. And so Chris made a variety of particles. One is just naked particles, and you can see that there's some uptake. The green is the particles, the blue is the nucleus. These are DIC images of the cells in culture. If you add antennapedia, or this penetrin peptide, you see much more abundant uptake. If you add folate, you also see more abundant uptake. And if you add them both together, you see more than you see with either one alone. This is even though quantitatively there's less penetrin and less folate on these particles than there were on either of these. And this can be blocked with folate. So this is an example of making a particle that is specific to cells, specific to cells that bear the folate receptor, and also enhanced for uptake because of the addition of penetrin. So what I hope I've showed you in a relatively short period of time is that particles like this can provide controlled release of many agents and at levels that are useful for treatment of tumors. And we think that there's clear pathways for translating these into clinical practice. That convection-enhanced delivery techniques allow us to place a dose of particles at the site at a defined location in the brain, and we're hoping that's going to be a useful approach for treatment of brain tumors. But further, I think surface modification, as we get a better handle on how to make these particles that are smart in very particular ways, might allow us to do even more. I tried to mention the students and fellows that were involved in this work as I went through it. Amaryllis Sanchez, who was a graduate student at Cornell, Tariq Fahmy, who many of you know, Andy Sawyer, a PhD student in chemical engineering, who is now a postdoc at Yale, Toral Patel, a fabulous neurosurgery resident, but I would guess all the neurosurgery residents here are fabulous, but Toral might be especially fabulous. Zhangbing Zhao, who the new father, who has made a dramatic progress in our ability to make particles that target tumors. Kim Woodrow, who's now an assistant professor at the University of Washington, and Chris Cheng, who's in the final stages of his PhD work in MBMB. Thanks, did I do?